Welcome to Project Lead the Way, Principles of Biomedical Science, Activity 3.1.1, Part 2 through Part 4. The first thing that I'm going to go over in this podcast is how to look at blood under microscopy or using microscopy. When you're looking at blood, the first thing that you should be able to see are your erythrocytes. And your erythrocytes naturally will appear a lighter pink color. You might even see or they might even look a little translucent, and so in our diagram right here you can see your erythrocytes or your red blood cells. The white here would represent where we would expect to see our plasma. So our plasma, once again, is that fluid in between our cells. The larger cells that you can see are nucleated. You also can see they appear a little bit darker in color. These are your white blood cells. And there's lots of different types of white blood cells that exist in our blood. We'll learn more about those when we get to infectious disease. And then over here, a little bit smaller than your red blood cells and darker in color are your platelets that will be involved in clotting and plugging up any sort of damage sustained to the circulatory system. When talking about sickle cell disease, the characteristic that's very unique in patients that suffer from sickle cell disease is a different shape of red blood cells. So when looking at this diagram here, you can see both regular red blood cells and sickle-shaped cells that appear more elongated and less round. When you're looking at blood and how it naturally flows through the blood vessels, being nice and round allows the blood vessels, or allows the red blood cells to travel through the blood vessels in an easier way. When we have abnormal or sickle-shaped red blood cells, it makes it more difficult for these red blood cells to travel through our blood vessels and puts you at increased risk of them getting stuck and or clotting, especially in areas where there are bends in our blood vessels, such as our joints. When discussing sickle cell disease, it sometimes can be called sickle cell anemia. And I wanted to go over what anemia actually is and how anemia is diagnosed. A hematocrit is a blood test that is used to evaluate the levels of red blood cells. So in the previous podcast, I went over what the normal value or percentage of red blood cells should be compared to the other components of the cell. To perform a hematocrit, you need to withdraw blood into a hematocrit tube, which is a small, generally glass vial, and then you will put that glass vial or that tube filled with blood into a centrifuge. And that centrifuge will separate the whole blood into plasma and your red blood cells. So looking at this diagram on the right, you can see a hematocrit tube that has been centrifuged. The plasma will be towards the top. Then you'll, at the bottom, see your red blood cells, and in between your red blood cells and your plasma is where you will find your buffy coat or your white blood cells and platelets. A hematocrit is generally number of a hematocrit, I should say, is generally a percentage. And so to calculate your hematocrit, you're going to take the red blood cell volume, divide it by the total blood volume, so including the plasma all the way up to the top of the value, and you'll multiply that by 100. And that will give you some sort of score or percentage. What's normal for a male is different than what's normal for a female. Normal values for a female are somewhere between 35 to 46%, whereas normal values for a male are somewhere between 42 to 54%. Anything below 42 for a male would be characterized as a low hematocrit. Anything lower than 35 for a female would be considered low as well. Anything higher than 54% for a male would be considered a high hematocrit reading. Anything higher than 46 would be considered a high hematocrit for a female. There's a couple things that could be the cause of a high hematocrit reading. First and foremost, dehydration. So if somebody comes in and they're super dehydrated, the amount of water that's in their plasma is going to be less. And since the hematocrit reading is the proportion of red blood cells to whole blood, if there's less water, there's less plasma. If there's less plasma, it could indirectly influence the ratio of red blood cells to whole blood. In addition, excessive red blood cell production, if there's something that's causing the body to produce too many red blood cells, that would result in a high hematocrit. Also, low availability of oxygen. So if the oxygen is not available, it's going to stimulate erythropoiesis, which will then increase the number of red blood cells present in the body. A low hematocrit score can be caused by a couple different reasons as well. First and foremost, any sort of bleeding. So if somebody's losing a lot of blood, either because of some puncture in an organ or external bleeding, that would result in a low hematocrit reading. In addition, premature destruction of red blood cells. So red blood cells are supposed to live 120 days. If there's something that's causing the red blood cells to not live that 100 days, that could then in turn reduce the number of red blood cells present in the bloodstream. That's something that's very characteristic of those sickle cells. The sickle cells do not last as long as normal cells. In addition, our third point, decreased production of red blood cells. There's a lot of ingredients that are required to make a red blood cell. Iron either through your diet, your body has to absorb that iron to make the hemoglobin so your red blood cells can be made. There's a lot of factors that can influence the whole production of red blood cell process, that whole erythropoiesis process. So if you're not consuming enough iron or your body's not absorbing enough iron, that could cause a decreased production of red blood cells, which in turn would then result in a low hematocrit. In addition to overhydration as our last point. As I mentioned before, sickle cells are destroyed faster than normal cells, which would result in a low hematocrit. The drug that was found in Anna's system is called hydroxyurea. Hydroxyurea is a medication that is used for patients that have sickle cell disease to prevent any sort of sickle cell crisis. And so sickle cell crisis are episodes of severe pains generally in the joints. And by taking this medication, it reduces the risk of those events aforementioned actually happening.